Okay, so I counted about 5 million cells, exactly. So that is hopefully what you got as well when you did your count, which is a ton of cells, which is too many for that flask, but we got there. So I should have passaged much earlier, probably yesterday. Okay, so I'm just cleaning this up, as you guys should always do. This is convenient. So I want to seed 50,000 cells in my new flask. That's what I've been doing, and it seemed to work well. So we can go ahead and, 500,000 cells per mil. I need 50,000 cells. That's an easy calculation. I just need 100 microliters of cell suspension in that new flask. So that's the first thing we'll do. We'll get this new flask out of the way. Sit well. Okay, and the next thing I need to do is I'm going to seed cells into my well plate. So I need to calculate how many cells I need to feed in there. So you guys should be able to do that calculation. I told you I wanted 10,000 cells in each well, right? And we're going to put 200 microliters in each well. And I'm going to do 2 well plates. So that means I have 120 wells I need to fill. So we could do the math and figure out how many cells you need. From my calculations, I think we need 30 mils total of cell suspension. That's giving us 250 microliters per cell, which is going to be, plenty. And we need it to be at 50,000 cells per mil. So it's, again, very convenient that we have 500,000 cells per mil from our flask. Making that 50,000 is just taking 1/10 of that. So if I take 3 mils, and so I'm going to make 30 mils total. So I'm going to take 3 mils from that, 27 mils from my low serum DMEM with no cells. That will give us all the cells we need in 30 mils. Okay, so 3 mils. I'm actually going to do it kind of accurately using this pipetter. I'll do it 3 times. But first I'll add the 27 of my low serum media. Now I'll go ahead and use a 25 mil pipette for that. And we'll go up to the -2 line. That was very awkward. My thing fell over. That's when you know you're getting too much trash. Okay, I'm going to make sure I label this before I screw things up. All right, I'm going to say this is 50,000 cells per mil or 96 well plates. Okay, I'm going to get 3 mils, as I said, of this. Again, you could probably get away with the 5 mil pipetter, serological pipetter for this, but might as well try to get an accurate number of cells here. So I'm just mixing around. Again, the cells will probably have settled while I was doing some counting. So I want to make sure I get a good homogeneous cell suspension. Okay. Again, 1 container opened at a time. I can reuse this because I haven't contaminated it all. I was very careful. Okay, so that gives us all of our cells, okay? These cells we don't need anymore. They're essentially trash. So I'm going to go ahead and stick those over there as well. Those are things we're going to need to bleach out. And so now we can get ready to see these 96 well plates. So I haven't actually labeled these well plates yet, so that's going to be something I'm going to want to do before too long. Just to be sure I'm doing things right, and don't want to, I don't want to erroneously put the wrong thing in. Okay, so the outside wells I'm going to fill with all PBS, right? And then I'm going to have 5 wells that are control 1. Okay, I'm just going to kind of put a line there, make sure we know. And control 2 is just normal. These will be bleached. Okay, and then I'm going to have experiment 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? So I'm just going to label these 1 to 10 because I have in my lab notebook what they're going to be. Okay, just make it clear that we're in this box area. Okay, and I'm going to go ahead and label the whole plate cytotoxicity assay plate 1. Right, and I know what that's going to be in my lab notebook. So what I'm going to do for this one is I'm going to do ethanol, and then I'm going to test different sugar concentrations. Okay? And then I'm going to do the same thing over here. Again, some of this labeling is just a sanity check for you. So I know where all my experiments are. I know where my controls are. Cytotoxicity assay plate 2. Okay, so in this one, I decided to test bleach to see how much bleach will take to kill cells in the media. And then also I want to test if antibiotics will have any effect. So we're going to test increasing antibiotic concentration. Right, so those are basically going to be our 4 little experiments we're going to do. Ethanol, sugar, bleach, and antibiotics. Okay, so this is where things start to get tricky as far as filling up this well plate. We could do each of these individually, which would be a really long time to fill up each of those. So we luckily have this thing called a multi-channel pipetter. So it has 1, 2, 3, 4, 5, 6, 7, 8 dispensers, and it's convenient for each of the 8 columns here. Unfortunately, we're not always going to be using 8, but you can just fill like 5 of these, right, and get the right number. So I'm going to go ahead and take a look here. Yeah, we got some tips rows that are only 5, and so that will work pretty well for us. Okay, but in order to fill this, right, we also need to use a special tool, which is this reservoir, for lack of a better word. So you put some liquid in there, and then you can set your, so we're going to be using 200 microliters. So let me go ahead and change this to 200. Okay, and it works just like a normal pipetter. But we'll have to suck up the liquid from here, and then we'll have to put it in the well plate. And this is going to be a real sterile challenge. You notice this stupid thing doesn't come with a lid for some reason, right? If I had packaged them, I'd maybe put a lid on there. So we have to be really careful about contaminating this. So that's where you really have to pay attention to hands. And so I'm going to start moving stuff out of the way over here to make sure I do my best at not contaminating things. The other thing is I know I'm going to need lots of tips, so I got this tip box as well I brought in. These are sterile, of course, that's why they're wrapped like this. Okay, so we're going to start with PBS. I've already calculated how much PBS I need to fill all these wells, right? There's going to be 8 plus 8 plus 10 plus 10, right? That's 36 in each plate, so I need 72 72 wells full of PBS. How are these things open? Not that way. These are sterile, that's why they're in these individual packages, because they have been sterilized at the factory. The trick is keeping them that way. You can be real careful with this. Alright, so as I said, there's 72 total wells. If I plan for like 250 microliters each, just to make sure I have enough, right, 72 divided by 4 is 18. I think that's right. So I need about 18 mLs of PBS. Okay, again, I'm going to be very careful, not going over this unnecessarily. So I'll probably just get like 20 mils of PBS just to be sure. And again, we're gonna do our best here, right? Like you can imagine, as we open this thing, it's gonna be really hard to keep things tracked. So I'm gonna grab 8, and these are the tips are conveniently located in 8 rows. So I just press down on all those. I have 8. Again, we already said this. Push it down. Suck up. Put in the other rows. All right, I haven't contaminated tips yet, so I'm going to go ahead and reuse them. I'm pretty good about this. And so this is where maybe we're a little bit careful here. All right, so we don't, like if I put in 8 there, that's going to be kind of painful. So I'm going to go put in 8 in these other ones. Yeah, I think I actually sucked up too hard that time. Nice and gentle. Okay, so I got everything except there's a total of 4 on each that I've missed. So I'm just going to do those manually. Be very careful about reaching around here so we don't go over. So you see I'm covering it every time I open it because my hand is definitely going over it, right? But as long as it's covered, I'm in good shape. Okay, so we got all the PBS in. This PBS is going to go away, so I'm going to put it all the way over here in the trash area. And now next we want to seed cells. So it's going to be the same thing where we're going to put the cells in this tray, trying our best not to contaminate them. Okay, as I said, I need 30 mils of cell suspension in there. So here's my cells. I'm going to mix them before I put them in there because they have been settling for a little bit. Everyone likes bubbles. Okay, so we're basically going to do these things 5 at a time. 200 microliters. I'm going to try to get 5 at a time here. So it does have 8 channels, but if I only pick up 5 pipette tips, that's how we can do 5 at a time. I'm going to tell you right now, this is not easy to me. It takes a lot of aim to make sure I'm putting them all in a well, make sure I'm not putting them into a well that already has PBS or does not already have cells in them, because that would be a little bit of a disaster. You know, I guess I could have done 6 at a time. Not sure what I was thinking here. I was thinking each group has 5, but now as I'm doing this, I realize I could have just done 6 and done the controls as well. Okay, I'm going to go ahead and get rid of these just so I can pause. Okay, so that well plate is done. And now I'm going to do this one here. This time I am going to do 6 at a time. It is important to get the same volume in each of the wells, because that way each of the wells gets seeded with the same number of cells. Okay, and so you can see I lost a pipette tip there. Okay, this is an interesting little semi-disaster that we're going to avoid by using our other pipette tip and pushing out, sucking up those bubbles. Okay, so that's okay. I think we survived that, but these are things that happen, right? And coming up with a contingency plan is important. And so what probably happened is I didn't provide equal pressure, so I'm making sure I get equal pressure on all those pipette tips so that they suck up and they dispense the exact same amount of liquid. Okay, so I did it. I'm feeling pretty relieved right now. I think I did a pretty good job without major issues or contamination or making a big disaster of everything. So we're going to clean all that up later. But right now I can take these 2 well plates and stick them into the incubator.